Introduction. Welcome, medical students, to today's lecture on cell biology. In this comprehensive script, we will delve into the fascinating world of immunology, specifically focusing on the complex processes involved in immune cell development, function, and response. We will discuss various topics in detail, starting with hematopoiesis and concluding with the concept of immune cell exhaustion. So let's dive right in and explore the intricate workings of the immune system. Hematopoiesis is the process of blood cell formation that is essential for the immune system. It happens in the bone marrow, where hematopoietic stem cells differentiate into different types of immune cells like T cells, B cells, natural killer cells, and myeloid cells. The differentiation steps are controlled by signaling pathways and transcription factors, ensuring the production of various immune cells to fight against pathogens. This video will provide an overview of T cell development, specifically focusing on the process of TCR rearrangement. We will discuss how T cells recognize antigens and the importance of successful TCR gene rearrangement for immune system function. B cells have a crucial role in adaptive immunity by producing antibodies that neutralize pathogens. The BCR recognizes antigens and triggers signaling events leading to B cell activation and antibody production. B cells also present antigens to helper T cells, initiating B cell activation and subsequent differentiation into plasma cells or memory B cells. During antigen presentation, immune cells called antigen-presenting cells, APCs, capture and process antigens. They then display fragments of these antigens on MHC molecules. There are two pathways, MHC class I for intracellular antigens and MHC class II for extracellular antigens. This process coordinates immune responses and directs specific immune functions. Co-stimulation and immune checkpoints are regulatory mechanisms that balance immune responses, preventing excessive activation or autoimmunity. Co-stimulatory molecules, such as CD28, on T cells interact with their ligands, such as CD80 and CD86, on APCs, amplifying T cell activation signals. Conversely, immune checkpoint molecules like CTLA4 and PD1 act as brakes on T cell activation. Their engagement with ligands on APCs leads to T cell inhibition, preventing excessive activation and tissue damage. Dysfunction or dysregulation of these checkpoints can contribute to immune-related diseases and can be targeted for immunotherapy. Cytokines are small protein molecules that mediate communication between immune cells and regulate immune responses. They play diverse roles, such as promoting cell proliferation, differentiation, migration, and immune cell activation. Cytokine signaling involves the interaction between cytokines and their specific receptors on target cells. This interaction triggers downstream signaling cascades, culminating in the activation of transcription factors and modulation of gene expression. Examples of cytokines include interleukins, interferons, and tumor necrosis factors, each with unique functions within the immune system. T cells can differentiate into distinct subsets with specialized functions, known as T cell polarization. This process is influenced by various cytokines and environmental factors. Th1 cells, for instance, secrete interferon gamma and play a crucial role in cellular immune responses against intracellular pathogens. Th2 cells, on the other hand, produce interleukins such as IL-4 and IL-5, orchestrating antibody-mediated immunity against parasites and allergens. Th17 cells contribute to immune defense at mucosal surfaces, while regulatory T cells, TREGs, maintain immune tolerance and prevent excessive inflammation. Effective immune responses require immune cells to migrate to infected or inflamed sites. Lymphocyte migration involves a highly coordinated series of events, including rolling, adhesion, extravasation, and tissue-specific homing. Selectins and integrins expressed on immune cells mediate their interaction with adhesion molecules present on endothelial cells lining blood vessels. Chemokines, a family of chemotactic cytokines, drive the directional movement of immune cells towards specific anatomical locations. These intricate processes ensure immune surveillance and the efficient targeting of immune responses to the sites of infection or tissue damage. Antibodies, also known as immunoglobulins, IG, are secreted by plasma cells and play a significant role in neutralizing pathogens. Antibody production is a complex process involving B cell activation, somatic hypermutation, and affinity maturation. Upon encountering an antigen, B cells differentiate into plasma cells, which secrete large quantities of antibodies specifically tailored to the antigen's epitope. Affinity maturation, facilitated by somatic hypermutation and clonal selection, improves the binding strength of antibodies to antigens over time, resulting in a more effective immune response. Interactions between T cells and APCs, particularly dendritic cells, are crucial for initiating and shaping immune responses. Dendritic cells capture antigens, process them, and present antigenic peptides to T cells, inducing their activation. T cell activation requires the engagement of both the TCR and co-stimulatory molecules, such as CD28. Upon activation, T cells can differentiate into effector cells, capable of eliminating infected cells or providing support to other immune cells. These intricate interactions and signals between T cells and APCs ensure effective immune surveillance and response. Memory immune response is a phenomenon in which the immune system generates a rapid and robust reaction upon re-exposure to a previously encountered pathogen. Memory cells, including memory B cells and memory T cells, are long-lived and possess enhanced effector functions compared to naive cells. Memory B cells quickly differentiate into plasma cells, enabling a rapid production of antibodies. Memory T cells exhibit increased cytotoxicity and cytokine secretion, providing a rapid defense mechanism against reinfections. The memory immune response lies at the core of immunization strategies and the development of vaccines. During chronic infections or cancer, immune cells can become functionally exhausted, losing their ability to eliminate pathogenic threats. Immune cell exhaustion is characterized by sustained antigen exposure and the upregulation of inhibitory receptors, such as PD-1 and TIM-3. Exhausted immune cells exhibit reduced effector functions and impaired cytokine production, leading to compromised immune surveillance. Understanding the mechanisms underlying immune cell exhaustion is vital for developing targeted therapies to reinvigorate the immune response against chronic infections and tumors. In this video, we have covered the development and activation of immune cells, exploring complex processes like hematopoiesis and immune cell exhaustion. Understanding these mechanisms helps develop new treatments for immune-related diseases. Good luck in your medical careers and keep exploring the fascinating field of immunology.